Hi, my name's Tori Buckles. This summer I had the privilege of working as a research assistant on the oil pad restoration project at the Sable Institute of Environmental Studies. I worked alongside my research partner David Rowley in the field, as well as writing a research paper that will further the restoration of vacant oil pads in northern Lower Michigan, with the help of Susan Wilderman and Executive Director of Sable, Fred Van Dyke. Gradual forest loss is part of an increasingly common process in forest ecosystems worldwide. An example of this pattern of forest loss caused by landscape-level habitat modification is pictured here on one of our sites. Forest fragmentation caused by the proliferation of oil pads has long-term effects because the vacated oil pads do not naturally reforest. A period of significant oil drilling in northern Lower Michigan, pictured here, has produced an increase in forest openings and associated forest fragmentation of habitat. The first commercial quantities of oil were drilled in Michigan in 1925. Since that time, approximately 60,000 oil and gas-related wells have been drilled in Michigan, leaving behind plots of land like the one you just saw. In the past, when oil and gas companies vacated oil pads, contracts with government organizations required only the removal of their extraction equipment. Reforestation efforts were not required as the sites were expected to naturally reforest. Today, energy companies are required to follow a vegetative restoration plan developed by the Michigan Department of Natural Resources. Although grasses and other herbaceous species established on these extraction sites most remain barren of woody vegetation up to 30 years after oil extraction has ceased. In order to address this problem, the Northern Michigan Well Site Restoration Partnership was formed between the DEQ, DNR, Huron Pines, and the Sable Institute. Our collective vision is to restore native plant structure and ecological function of well sites and associated infrastructure while providing ecological, cultural, and economic benefits on a landscape scale. We studied 9 site locations in Antrim, Kalkaska, and Otsego counties. The Sable Institute, indicated by the blue star in the figure on the right, is located in the heart of the drilling and extraction in northern Lower Michigan. Our location gives us the unique opportunity to study a problem that directly impacts our local community. Our sites range from half a hectare to 1 hectare in size. In the image on the left, you can see the forest edges caused by fragmentation. The sites are open with no slope, and there is little topsoil, and the soil that is present is sand-dominated. Every site contains a 50 by 50 meter experimental plot. The plots are divided into 16 10 by 10 meter cells, each containing 1 surface treatment and 1 tree species. We implemented 3 active soil surface treatments: fertilizer and disking. We also used a control treatment where each tree species was planted in soil that had not been treated. 4 native species were used: white oak, red oak, jack pine, and red pine. These were chosen because of their ongoing use in the MDNR's forest management policy. We chose not to use early successional tree species because the goal of this study was not to recreate succession, but rather investigate the quickest methods to reforest vacant oil pads. In 2015, 900 trees of each of the 4 selected species were planted for a total of 3,600 trees. There were 225 trees for each species treatment interaction. For example, there were a total of 225 red pines planted in disked soil. Finally, each 50 by 50 meter plot contained 400 trees, and each 10 by 10 meter cell was planted with 25 trees. Soil samples were collected from 2015 to 2017 using a 15 centimeter soil collecting tube in a random fashion along 8 transects intersecting all treatments and tree species. Samples of forest soil were collected from adjacent forests at each pad along an interior forest transect. These were sent to the Soil and Plant Nutrient Laboratory of Michigan State University for analysis of macronutrients, micronutrients, soil composition, and organic matter. We also submitted soil samples to FiberTech Environmental Services for determination and presence of concentration of diesel-range organic compounds and heavy metals associated with oil drilling activity. We sampled vegetation using a Daubenmeyer frame at all sites along 8 50-meter transects placed in a systematic, random fashion intersecting each treatment and tree species. Vegetative cover was determined on a 0 to 9 scale within the frame. We determined density of woody vegetation in a plot by recording all woody plants identified as species within 1 meter of either side of each transect. Growth and survivorship of planted trees were recorded in June, July, and August from 2015 to 2018. When planted, each tree received a metal tag indicating the site, species, treatment, and individual. Plots were searched visually, and when the search became increasingly difficult, as the young trees became surrounded with vegetation, we used a metal detector to ensure the detection of all trees present. We answered these fundamental questions. What differences in soil characteristics exist between oil pads and adjacent forests? What surface treatments are most effective at enhancing survivorship and growth of planted trees on vacant oil pad sites? Which native tree species would demonstrate highest survivorship and growth when planted on vacant oil pads? As well as, which management treatments associated with reforestation best reduce presence of non-native species? And finally, how do surface treatments influence composition of herbaceous and woody growth? Using these 11 analytes, we determined that variance in soil characteristics was primarily due to differences in composition between oil pad sites and adjacent forests. We refer to this as location, and it is illustrated by the orange columns in the figure. You'll notice for the first 8 analytes, the variance attributed to location was equal to or greater than 50%. Only 2 analytes, zinc and chlorine, vary due to differences between individual oil pad sites, illustrated by the blue columns on the right of the figure. Using these results, we were able to identify where limiting factors in the soil characteristics might have been present. We sampled for 9 toxic heavy metals: arsenic, barium, cadmium, chromium, copper, lead, mercury, selenium, and silver, as well as diesel range organics. Only chromium and copper differed between soils on oil pads and forests. Concentration of both metals were higher on oil pads, but the associated values fell within normal soil ranges as established by the Agency for Toxic Substances and Disease Registry, as well as the background soil survey produced by the MDEQ in 2015. The concentrations of the remaining heavy metals were either undetectable or fell within normal soil ranges. Based on this, we concluded that lack of reforestation was not due to soil toxicity. The abiotic factors, pH and organic matter, differed between oil pads and forests. The pH of our oil pads was more alkaline than forests, and this finding was consistent with other investigations that examined similar differences. Oaks and pines are adapted to grow in acidic soils. In alkaline soils, cationic nutrients are less soluble and therefore less available for absorption and metabolization. Without these nutrients, growth and development may be stunted in oaks and pines. Low levels of organic matter also limit tree growth. Organic matter acts as a sponge which collects and retains soil moisture. Without adequate amounts of moisture, competition between trees and herbaceous plants increases. We sampled for 7 macro and micronutrients: potassium, calcium, magnesium, zinc, manganese, iron, and chlorine. 3 of these, calcium, magnesium, and iron, differ between pads and forests. Concentrations of calcium and magnesium were higher on pads than forests and were not limiting to growth. On the other hand, we encountered iron-deficient soils on pads. Iron deficiency can induce iron chlorosis, which lowers the rate of photosynthesis, turns tree leaves brown, and can ultimately cause death. Oaks are especially susceptible to chlorosis. Returning to our first fundamental question, 2 toxic heavy metals, 3 nutrients, pH, and organic matter differed between oil pads and forests. Each section displayed here represents a species of tree that was planted in 2015. You'll notice a decrease in survivorship from the time of the initial planting and a stabilization after year 2017. Disking, indicated by the gray bar, produced the highest survivorship in every species and in every year. You'll notice that the species with the highest survivorship was red pine, indicated in the last section. Fertilization, indicated by the blue bars here, resulted in the lowest survivorship in every species in every year. Disking enhanced the survivorship of planted trees. Disking reduces competition by removing existing herbaceous vegetation. It also stimulates early root growth in seedlings by aerating and breaking up the soil, allowing the roots to expand. To answer our second fundamental question, disking was the most effective treatment, enhancing both survivorship and height. Trees survived better in the non-treated control section than in the fertilized treatment, indicating that fertilizer actually suppressed growth by increasing competition with herbaceous vegetation. Pines were shown to experience positive growth in all surface treatments. In contrast, oaks experienced reduced height in all treatments. Red pine in the disc treatment experienced the most overall increase in height of all tree species, indicated by the tallest yellow bar. Jack pine experienced the greatest increase in height in the fertilizer and disc treatments, indicated by the tallest gray bar. To answer our third fundamental question, red pine had the highest survivorship and overall growth in the disc treatment, while jack pine had the highest growth in fertilizer and disking. Oaks were shown to decrease in height in subsequent years due to the effects of herbivory by white-tailed deer. Herbivory was an overriding determinant of treatment upon oaks. Protection, such as shelters or mesh barriers, might have increased survivorship and growth of oaks. Using tree shelters has been shown to decrease herbivory from 68% to 4% in red oaks. Oaks grown in mesh barriers were 10% larger than oaks in unprotected sites. Recall that because vacant oil pads do not naturally reforest, non-native species do not compete with trees or nutrients. Because of this, non-natives spread quickly to fragmented forest sites. The negative effects of non-natives are well documented, and all 9 of our sites suffered from invasion of multiple non-native species. Here are the 3 most common non-native swarms on our sites: St. John's wort, spotted knapweed, and lippy spurge. In the 3 active surface treatments, fertilizer with disking, and disking, non-natives accounted for 52 to 61% of the total plant cover indicated by the green portion in each of the first 3 columns. Only the untreated control plot had a greater portion of native species. Because of the proportion of native to non-natives in treatment plots was greater than the control plot, it is clear that active treatments did not reduce the spread of non-natives. This answers our 4th fundamental question. Richness and diversity of the herbaceous community did not differ depending on surface treatment, answering our 5th fundamental question. Now, on a small portion of Site 3, we observed an unexpected colonization of volunteer or unplanted jack pine, and a dramatic increase in the height of planted jack pine after disking was applied in 2015. This portion of the site had previously been used as a logging deck to stack felled trees during active operation. When we selected this site for study, there was no deck and no evidence of reforestation. However, surface litter and wood chips were present and were mixed into the soil when disking was applied. Eldridge and his colleagues may have provided an explanation for this. Wood chips have been found to increase organic matter on nutrient-poor soil. Increasing organic matter led to an increase in soil matter, which reduced competition between trees and plants. In addition, the investigators found wood chips can actually reduce the presence and abundance of non-native species. Forest managers who want to most rapidly mitigate the problem of site-specific deforestation associated with vacated oil pads should plant red pine on disked soil and avoid fertilizer if used with our selected tree species. For the research that examined the survivorship of tube or mesh-protected oak seedlings compared to unprotected oak seedlings on selected oil pads, and includes surface treatment of residue and wood chips mixed with disked soil. I'd like to thank Fred Van Dyke for his guidance, Susan Wilderman for her oversight and assistance with fieldwork and data analysis, the MDNR for their ongoing cooperation with our research, and Shell Oil and ConocoPhillips for providing the funding for this research, as well as my research partner, David Rowley. Thank you.